Yo, welcome back to the deep dive. मैं हूं आरव, प्रिया मैम is here, मृणाल and दिव्यम वापस आ गए.
We're basically recurring characters at this point. Like the post-credit scene nobody skips.
और आज का टॉपिक मुझे बहुत पसंद है, डीएनए रेप्लिकेशन. ये तो मॉलिक्यूलर बायोलॉजी का फाउंडेशन है.
Divyam, I love the energy. So yes, DNA replication. This is the process that allows one cell to become two identical cells. Every time your skin heals, every time your hair grows, every time your gut lining replaces itself, replication is happening.
मैम नीट में इसका क्या सीन है? Like real talk, कितने questions आते हैं?
देखो, replication is a 3 to 4 question gold mine every year. Enzymes, direction of synthesis, Okazaki fragments, Mendelssohn-Stahl experiment. NEET loves asking tricky MCQs on this. And DW, they'll give you a 3-hour lecture where you still won't know the difference between leading and lagging strand.
Honestly, DW's replication lecture has the same energy as Windows Update. You're stuck watching it, it takes forever, and when it's done, you're not even sure what changed.
भाई, और डून एकेडमी का तो एनिमेटेड वीडियो होगा ना? डीएनए हेलिकेस को सुपर हीरो बना के हेलिकेस मैन अनवाइंड्स द ट्रूथ.
Stop. That's actually probably what they do. Okay, focus. Let's do this properly. DNA replication in 10 minutes. Enzymes, mechanism, Mendelssohn-Stahl, NEET traps. सब कवर होगा.
तो मैम पहले बताइए डीएनए रेप्लिकेशन में कौन-कौन इन्वॉल्व है? Like the cast of characters.
Think of it like a highway construction project. You need specific workers for specific jobs. पहला वर्कर DNA Helicase. ये वो गाय है जो जिपर खोलने का काम करता है. डबल हेलिक्स को अनवाइंड करके 2 सिंगल स्ट्रैंड्स बनाता है.
So Helicase is essentially the guy who unzips your jacket. Except the jacket is 3 billion base pairs long.
Good analogy. अब जब हेलिकेज unwinds, the point where the 2 strands separate is called the replication fork. Picture a Y shape. That's your fork.
अरे ये तो वैसे है जैसे दिल्ली मेट्रो का ट्रैक डिवाइड होता है ना. राजीव चौक पे एक लाइन 2 डायरेक्शंस में जाती है, ब्लू लाइन और येलो लाइन.
Nice one, Divyam. That Y junction, that splitting point, that's your replication fork. अब next player, single strand binding proteins या SSB proteins. जब helicase unwinds करता है, the separated strands want to snap back together. SSB proteins hold them apart. Think of them like bouncers at a horse-cast bar holding the door open.
And then there's Topoisomerase. This one's important. When Helicase unwinds DNA downstream, it creates tension ahead, like positive supercoils. Topoisomerase relieves that tension by cutting, rotating, and resealing the strand.
Exactly, Mrinal. NEET trap alert. Topoisomerase is not the same as helicase. Helicase unwinds. Topoisomerase removes the tension caused by unwinding. 2 different jobs.
Okay, तो हेलिकेस opens, SSB holds, topoisomerase relieves tension. अब actual copying कौन करता है?
DNA Polymerase 3. This is the main enzyme. ये new strand synthesize करता है by adding nucleotides complementary to the template strand. But, and this is critical, DNA polymerase 3 can only add nucleotides in the 5' to 3' direction. It cannot start a new chain on its own. It needs a primer.
प्रीवर? वो पेट वाला?
No, not wall primer, RNA primer. It's a short stretch of RNA laid down by an enzyme called Primase. DNA polymerase needs this little RNA starter to begin adding DNA nucleotides. Like how you can't start a Swiggy order without adding your address first?
Mnemonic time. Remember the enzyme squad? HPPPL. Helicase unwinds, primase primes, polymerase 3 builds, polymerase 1 replaces primers, ligase seals. Say it. Happy people play polo lazily.
Happy people play polo lazily. भाई ये याद रह जाएगा.
Okay मैम, अब actual process. डीएनए रेप्लिकेशन कैसे स्टार्ट होता है?
First, you need an origin of replication. In prokaryotes like E. coli, there's one origin called ORI-C. In eukaryotes, there are multiple origins because the DNA is too long for one starting point. Imagine अगर एक ही Blinkit warehouse से पूरा दिल्ली deliver करना हो. Impossible. You need multiple warehouses. That's why eukaryotes have multiple origins.
That's actually a solid logistics analogy. Multiple origins of replication are essentially parallel processing, like having 20 tabs open instead of loading one page at a time.
Exactly. From each origin, two replication forks move in opposite directions. This is called bidirectional replication. NEET tests this. Replication is bidirectional from the origin. True statement.
ओके तो फोक बना, हेलिकेस ने ओपन किया. अब क्या?
Primase lays down a short RNA primer on each template strand. Then DNA Polymerase 3 starts adding nucleotides. Now here's the key concept. DNA Polymerase 3 can only synthesize in the 5' to 3' direction. Only. No exceptions. No reverse gear.
लेकिन मैम डीएनए के दोनों स्ट्रैंड्स तो एंटी पैरेलल है ना. एक 3' तो 5' डायरेक्शन में है, दूसरा 5' तो 3' में.
Bingo, Divyam. Yahi to problem hai. The template strand that runs 3' to 5' is easy. DNA polymerase reads it 3' to 5' and builds the new strand 5' to 3' smoothly, continuously towards the fork. This strand is called the leading strand.
The leading strand is basically the kid who finishes the exam paper start to finish without looking up. No drama, no complications, just continuous synthesis.
Exactly. One primer, continuous synthesis, smooth ride. The leading strand is the IIT topper of replication.
IIT टॉपर और दूसरा स्ट्रैंड?
That's where the chaos begins. So the other template strand runs 5' to 3', but polymerase can only build 5' to 3', so it has to synthesize away from the replication fork. मतलब direction उल्टी है.
मैम तो वो सिंथेसाइज कैसे करेगा अगर डायरेक्शन ही रॉन्ग है?
It does it in short fragments, discontinuously. थोड़ा synthesize करो, रुको, आगे जाओ, फिर से थोड़ा synthesize करो. These short fragments are called Okazaki fragments, named after Reiji Okazaki, who discovered them.
So the lagging strand is like writing an essay backwards, paragraph by paragraph, and then stitching them together. Each paragraph is an Okazaki fragment.
Perfect analogy. Ab sun lo carefully. Each Okazaki fragment needs its own RNA primer, so Primase has to lay down multiple primers on the lagging strand. Polymerase 3 extends each primer into a fragment. Then, when the next fragment bumps into the previous one's primer,
तो आरएनए प्राइमर का क्या होता है?
DNA Polymerase 1 comes in. Its job? Remove the RNA primer and replace it with DNA nucleotides. Basically, janitorial duty. Clean up after primase. Primer हटाओ, DNA लगाओ.
अरे ये तो वैसे है जैसे कंस्ट्रक्शन साइट पे टेम्पररी स्कैफोल्डिंग लगाते हैं ना. काम हो गया तो स्कैफोल्डिंग हटा के परमानेंट वॉल लगा देते हैं.
Exactly. RNA primer is the scaffolding. DNA polymerase 1 is the cleanup crew. Final step. After polymerase 1 fills the gaps, you still have nicks in the sugar phosphate backbone. Small breaks between fragments. Who seals them?
DNA Ligase, the molecular glue gun, joins the Okazaki fragments into one continuous strand.
Yes, mnemonic for lagging strand sequence. Priya orders pizza, lights go. Primase makes primer, Okazaki fragment extends, polymerase 1 removes primer, Ligase seals gaps.
प्रिया ऑर्डर्स पिज्जा, लाइट्स को. मैम ये आपके नाम पे है?
Marketing strategy, Arav. Ab NEET trap. Which enzyme seals Okazaki fragments? Answer: Ligase, not polymerase. Students confuse this all the time. Polymerase 1 replaces primers. Ligase seals nicks. Two different jobs.
Also worth noting, Okazaki fragments in prokaryotes are roughly 1,000 to 2,000 nucleotides. In eukaryotes, about 100 to 200. NEET has asked this size difference.
Good catch. Lock that in.
मैम अब वो एक्सपेरिमेंट मेसेल्सन स्टॉल नीट में हर साल आता है ना?
Every year. So 1958, Matthew Meselson and Franklin Stahl designed the most elegant experiment in molecular biology. They wanted to test how DNA replicates. There were three hypotheses: conservative, semi-conservative, and dispersive.
Conservative means the parent DNA stays fully intact and the daughter is entirely new. Semi-conservative means each daughter gets one old strand and one new strand. Dispersive means old and new DNA are mixed randomly within each strand.
Exactly. So what did they do? They grew E. coli in heavy nitrogen, N15, for many generations. All the DNA became heavy. Then they shifted the bacteria to light nitrogen, N14, medium, and allowed one round of replication. They extracted DNA and ran it on a cesium chloride density gradient centrifuge.
और रिजल्ट क्या आया?
After one generation, all DNA was intermediate density, one band. This ruled out conservative replication, because if it were conservative, you'd see two bands, one heavy, one light.
But intermediate density is consistent with both semi-conservative and dispersive. So they needed generation 2.
Exactly. After two generations, they saw two bands, one intermediate, one light. This confirmed semi-conservative replication and ruled out dispersive, because dispersive would give all intermediate bands of gradually decreasing density.
तो सेमी कंजर्वेटिव प्रूव्ड हो गया. ईच डॉटर डीएनए हैज वन ओल्ड स्ट्रैंड, वन न्यू स्ट्रैंड.
Neat trap. After how many generations can you distinguish semi-conservative from dispersive? Answer: 2 generations. 1 generation only rules out conservative. Lock this in.
There's also the proofreading angle. DNA polymerase has 3' to 5' exonuclease activity, meaning if it adds the wrong nucleotide, it can reverse, remove the mistake and try again. Error rate is roughly 1 in 10 to the power 9. That's one mistake per billion base pairs.
Mrinal, excellent point. This is the silent factor. DNA replication isn't just fast, it's accurate. The error rate with proofreading is insanely low. Compare that to RNA polymerase in transcription, which has no proofreading. That's why DNA is more stable than RNA. Your genetic library has a spell checker. Your transcription photocopies don't.
भाई, तो DNA polymerase में built-in Grammarly है. RNA polymerase free version पे चल रहा है.
That's the best comparison I've heard this month. DNA polymerase is Grammarly Premium. RNA polymerase is the free version with ads. Also, NEET asks, which enzyme has proofreading ability? DNA polymerase, not ligase, not primase. DNA polymerase.
Alright, summary time. Ma'am, 30-second lockdown.
Done. Replication starts at origin of replication. Helicase unwinds, topoisomerase relieves tension, SSB holds strands apart, primase lays RNA primers, DNA polymerase 3 builds new strand, 5' to 3' only. Leading strand, continuous, 1 primer, smooth. Lagging strand, discontinuous, multiple primers, Okazaki fragments. Polymerase 1 removes primers, fills gaps with DNA. Ligase seals the nicks. Meselson-Stahl proved semi-conservative using N15 and N14. Generation 1 rules out conservative. Generation 2 confirms semi-conservative. Proofreading by polymerase keeps error rate at 1 in a billion.
NEET traps, rapid fire.
5 traps. 1. Direction of DNA synthesis, always 5' to 3', no exceptions. 2. Which strand is synthesized continuously? Leading strand, not lagging. 3. Okazaki fragments are found on which strand? Lagging strand only. 4. Which enzyme removes RNA primer? DNA polymerase 1, not polymerase 3. 5. Meselson-Stahl used which organism? E. coli, not yeast, not Drosophila, E. coli.
One more bonus trap. Replication is semi-conservative and bidirectional. Both words matter. Semi-conservative tells you how strands are distributed. Bidirectional tells you the forks move in both directions from the origin.
और mnemonics याद रखना. Happy people play polo lazily for enzymes. Priya orders pizza, lights go for lagging strand. Done.
Students, DW will take 4 hours and still confuse leading with lagging. Dun Academy will animate Helicase as a superhero. Seep Ma'am, the self-proclaimed goddess of biology, will probably have you chant the enzyme names like mantras. We gave you logic, mnemonics, and mechanism in 10 minutes.
Subscribe, share with your NEET batch, drop comments. This was the Deep Dive Summit Neuro. Next episode, genetic code and mutations. Because apparently, one wrong letter can change your entire protein.
One typo. That's all it takes. Sickle cell anemia is literally a spelling mistake.
एक बॉस चेंज, पूरा प्रोटीन बदल गया. बायोलॉजी का बटरफ्लाई इफेक्ट.